Kaiser. So it's my pleasure to now introduce Dr. Ying Zhu to you. Dr. Zhu is currently based in Genentech, but was previously working at Pacific Northwest National Labs for almost 7 years, working on a lot of different analytical chemistry approaches, finally culminating in the co-invention of the nanopots technology that I'm sure we'll hear about. So I'm really excited as a fellow proteomics researcher to really learn more about how we can do single cell proteomics. So thank you, Ying, and you may start your talk now.
Can you hear me well?
Yes.
Excellent. Thank you, Devang, for your introduction, and also I appreciate the invitation for me to. I'm not a plant scientist, so most of my research focus on technology development. I'm a chemist. So most of my research is focused on technology. So also because previously working on Pacific National Lab, and it's in Amazon, it's a user facility. We focus on provide different kind of capability to user to start the plant system. So before that, so here is my topic to introduce different technology we have tried to developing single cell proteomics and spatial proteomics for plant biology. So I want to give all the credit to my wonderful team back in PNL. So we developing the technology with Ryan Kelly and Liliana Katholieke to enabling the measuring small number of cells and use mass spectrometry. And most of my plant work is to collaborate with Amir Akami in the lab. And here is a postdoc and also early stage scientists working with me to generate the data to optimize the protocols. So also we also collaborate with other team members in PNL in single cell proteomics and spatial proteomics. And also we get a lot of collaborations with external vendors and to help us to optimize the system to analyze the data and to get the plant samples. So as you see today, most of existing single cell omics technology based on RNA sequencing, and it's well optimized, have very powerful, can run thousand single cells. But really, do we really need other omic technology like proteins? So we know most of the function effort and factor in all the mammalian or plant system is actually the protein. And what we try to do in the RNA sequencing is try to using the RNA abundant to inferring the protein abundant, because there's in theory that should have a good correlation. But really, we do a lot of studies have showing, and the correlation is not great. And it's understandable because both RNA and protein, they have different generation degradation rate. So, and also after you translate into the protein, that's auto-regulated by a lot of different mechanisms in post-transcription regulations. And because we do the measurement noise, so no matter we measure protein abundance or measure RNA, it's not precise. And also MRI is not very good and to predict very specific protein like extracellular matrix protein or long life proteins. And another why we use mass spectrometry other than antibodies or gene fusion approach. At first, we can, for plant proteins, we know that not many available antibodies for us to use, and the specificity of the antibody is not well known. We really don't know if the measurement is reliable. And if we use mass spectrometry, we don't have to limit to several target protein like GMPM cherry labeled protein, and we can measure all the protein present in the cells. So we can directly measure the cell from native plant without any gene manipulations. And now we already achieve 1,000 protein in single cells. So we start from the technology, we call it nanopods. It's also a microfluidics technology, but in different way. So we do not generate droplet in oil. What we do is that we print single droplet on the slides. So what we do is we sort single cell in the small wells and then use a robot to put the nanoliter liquid to do the protein extraction and digestion. So we get the peptide for the mass spectrometry analysis. So we have to make a robot to do all these liquid manipulations. So everything done in one pod. That we call it nanopod, because if we do the transfer, we lost more samples, more protein. And this is also very different because for protein, you have nowhere to amplify. So what you have is what you have in the cells. That we need high sensitive, high recovery technology to do the measurement. Is frozen again, apologize. Yeah.
Yes, I see your slides.
I'll try to do this. I don't know, it's because the Zoom or because the PowerPoint. So that's what we learn while we have to use the microfluidic technology. So because we do everything in small scale, we can reduce sample loss to get high sample recovery. It's especially for protein because protein is so sticky. And also, it's very important to realize if we do everything in small droplet, we have high enzymatic digestion efficiency and chemical labeling efficiency. Also, some practical issue is using small volume, we can reduce the contamination, improve the robustness of mass spectrometry. We can measure more cells. And then use microfluidics, we can do parallel sample preparation and high throughput analysis. So here is a very early stage. We try to coupling flow cytometry or other cell sorting technology to nanopod because it's open chip design. So we can actually put the chip inside of the FACS. So you can align to put the 1 cell or 3 cell, 10 cell into the flow cytometry. Here, if you look and see here, is not planned. So it's a HeLa cell. So just the demonstration of the technology. So we can, very early stage of the development, we can measure about 600 protein and among 300 common in all the single cells. So another direction technology space we are working on is coupling another single cell proteomics technology is SCOPE-MS. So it's approach using commercial isotopic tag and to tag each cell with isotopic. So we can measure, because mass spec measure the mass, so we can see the difference of different cells. Use the we call it report ions. Actually, it's a kind of chemical labeled with different isotopes. So we can measure them with mass spectrometry. So use the existing TMT tags. In theory, we can measure 16 single cells in one LC-MS run. So you can increase the throughput of the proteomic measurement. And but the challenge is the operation is very complicated because we put a single cell in 34-well plate, and finally we have to do everything combined together into a tube to do LC-MS analysis. So the throughput is quite limited. So that's we developing, because we can do microfluidic design. So we develop the next nanopod chip. So here is the chip we see is actually it's a cluster of small nanowires in small rings. So what we do is we do each TMT set labeling in one small clusters. And we have use different version of TMT. We can do 9 plex in one clusters, or use the new version, we can do 16 single cells in one clusters. So we can run over 500 cells in one microfluidic chip. So here we do, because it's the new generation, we actually do not use our homemade robot to do all the sample prep and cell sorting. We use a commercial system. It's called the Cello One, and it's used for some RNA-seq study also. So here we can use this system to do not only cell sorting, also sample prep. So what we do here is we sort single cell, use cell1, and then do the liquid pipetting and do the TM labeling. After TM labeling, we can just put a big droplet to combine all the different cells together in one TMD set to run them. Here also quickly to show how we can use the new N2 chip to see the difference. This is a PCA plot of the about 100 single cells. We can quantify about 1,500 protein in all the cells, and quite exciting to see so many proteins in single cell. So next, we are trying to move to the plant. So here we also start from a model plant, Acrobidopsis. So we use available protocols. It's generate a proplaster from the leaf, mesophyll proplasters. It's relatively pure. Use this approach. Is use a tap to remove most of epidermis, and so we can only sampling the center cells from the plant from the leaf. Here we can get a pretty good quality of the proplasters. So the cell1 allow us to sort different number of cells, proplasters in our nanopod device. The reason we use cell1 also capture each imaging during cell sorting. So if we see some cell interesting or some cell is outlier, we can go back to see if the some cell is different shape or different structure. That's very nice for us to troubleshooting also to see not only the omics measurement, also the imaging of the cell. Here is quickly to show you what we can see, how many protein, how many peptide we can detect from the single protoplasters. And here is 0 cells, and here is 10 cells, 3 cells, 1 cell. So we can get about 2,000 protein from 10 protoplasters and about 1,800 protein from single protoplasters. And we can see the quite stable measurement. The CV is actually the protein abundance. CV for 10 cells is about 20%. And from 1 cell is a little bit higher, but also pretty stable over all the protein in the proplasters. So if we do the PCA, and so we totally measure at protoplaster, as you can see here, use label free. So we can see two very clear outlier of the D4 and D5. And we really don't know why these two outlier. In theory, we should get the very similar mesophyll protoplaster from the leaf. So we go back to see the images as kind of different shape and also kind of like a fragment of the cell. So when we look at what the difference of this, actually is chloroplasters, and because we also find that this other challenging of to do the plant single cell, because cell is plant is so easy to fragment, to easily broken during the cell sorting, during sample prep. And a lot of we have dropped out of this. And also the cellular component analysis also tell us, you see most of this outlier actually is the broken cell. Majority of this fragment is chloroplasters. Here, we next want to ask if we can use single cell proteomics to identify, to see the drought induced or the other environment stress induce the protein changes. As you can see, what we do is we generate the same plant, same mesophyll proplasters, and from the plant under the control is 100% of the watering, and also we have different stress in low drought is 55% of the relative water content, and the moderate is 40% is more drought. Here is how many protein we can detect from single protoplasters. About in area about 2,000 protein from each cells. And here is we did a PCA projection, and you can see here the low drought do not introduce a lot of changes. I can see from the control low and the drought low, and we can see clear separation, PCA separation of the moderate drought between control and drought. And Also, we can see not all the cells exhibit the same response to the drought. Some have a high response, some had low, and we can see large change. So we can do the next stage analysis is do the look at what protein really change, which protein upregulated in drought stress cells. What's the protein or pathway downregulated in the drought stress leaf. It's not surprising in the upregulated. The first one is of the gene quality analysis is a response to water deprivation. It's clearly what we should see here. And also we see increase of the photosynthesis and duration precursor metabolites or energy. It also tells us the leaf start to increase the photosynthesis and also preserve a lot of energy for its response to drought. Also see a lot of catalytic activity increase. For downregulated one, we can see a lot of protein related to the translation and structure modeling activity. It's downregulated. It tells us the plant leaf try to reduce the protein synthesis to conserve energy, and maybe also change the structures to preserve the water. So we do get pretty good data from single cell proteomics of the leaf, and but we do have a lot of challenges working with the single pluripolar. At first, is very low rate of the single cell suspension from the plant. That's very different from mammalian. Also, as we see we really want to understand what's the trajectory of the development of the different plant cells. If we digest everything, it's very hard to take back because we lost all the parasitic information of the cell. And also we also know after digest the cell wall, we lost all the cell wall proteins. And also cell security protein, you also cannot lose to them because they are not inside the proplatters. And also, if you want to integrate with the imaging technology, it's better with the imaging. That's why at the very early stage of our development, we try to coupling the nanopod with LCM. So let the capital make dissection. So that actually is a imaging technology coupled with the laser ablation. So what we use is the light microscope, is that time. So this one allows us to direct coupling our nanopod chip to the LCM systems. So we can do the cell isolation. Here is the movie, hope it's working. So to do like as small as 20 micron resolution dissection, and then collect into the nanopod. As you can see, we can, depending on how many cells you want to collect, you can do 1 cell, you can do like 100 cells. There's no upper limitation. So we also start developing the technology from the mammalian tissues. Here is actually the human pancreas tissue, because we know the pancreas have a function unit, it's islet. So we can draw grid across the islet. So we can analyze each pixels. And so we can generate a heat map across the entire islet. So understand the microenvironment of a single islet. So we can intercut. Even we have 50 resolution, actually that's a 5 to 10 cell, because mammalian cell quite small. And here is we can get from the single cut, we call single pixel, we can get 23,000 peptide from each pixels. And for protein, we can get over 3,500 proteins. And it's here I try to show, because we have this heat map, and also quantify over 3,000 protein. We can do a lot of spatial omics analysis. Here we use spatial network analysis, try to looking for different patterns across this islet microenvironment. So we can get 3 different major clusters. It's showing the RLED proteins and also acinar proteins. And also we can see some, gradient across the entire section, no matter its cell type. Likely here is top-down gradient, like usually the nutrition gradient across the tissues. So working with the plant, so here we start also try to move the technology from mammalian system to plant system. So we working with the popular, and Amazon we are environmental research, and we focus on a lot of biology, bioenergy, related to the plant system. So the popular, that's why we have a special interest to popular. So we harvest the leaf, we develop protocol embedding them in the tissues, in the CMC blocks. So we do the sectioning, we use LCM because we can cut section, we can know exactly what we are looking for. We cut them into nanopod, analyze with nanopod LCMS. So here we select 2 different tissue, and one is the leaf. So for the leaf, we select the mesophyll and palisade and vasculature. For the root, we can also select epidermis and stellar and the root context. So what we focus on is vasculature and the context. As you can see, we do not get as high as the single cells, the cover in terms of the coverage, because that's some different in terms of the cell tissue treatment. So, but still we can get a pretty good coverage, but over 2,000, above to the 2,000 protein across different tissue, like leaf, we can get totally 2,600 protein. There's some common protein between palisade and the vasculature. Similarly, we can get similar coverage for the root. And we can a lot of common protein between them. The more protein from the vasculatures. So just a quick analysis of the different cell type specific, because we know what we are looking for in this, so we can quickly generate the cell type specific protein and locate what's the function different of this the cell types. So for the leaf, we can see, for the mesophyll or palisade cells, we can see the most of protein involved in the photosynthesis because they are leaf. And for the vasculature, we can see most of cells is involved in the sugar one synthesis, it's energy related. And for the root, we can see a lot of, for the root contact, we see also a lot of sugar by synthesized protein and phenolic compounds enrich. And for the vasculature, we can see more the pathway related amino acid synthesis. It's quite encouraging to see use the technology. We can see clearly what we are looking for in the imaging. Isolate for the omics, you can see more protein than the imaging approach. You can combine them together and to get more, more detailed information of the different cell type. So for the summary, so we can show we have all the technology to measure not only single cells, get oversized protein from single protons, and also get the cell type specific protons use the LCM coupling with the technology. So also we have data to show we can measure the proton regulation response to different stress. And here we only show the drought stress, but in theory also can apply to other stress to understand how the plant responds to the environmental. Most exciting is because we are funded by the DOE user facility, so most of the technology we present here is available through Amazon user program. So you can contact with our colleague in the Amazon, Amir, Akami, and Lilian Katholieke if you want to try the technology present here. So here is all the funding agency. Apologize for the problem in technical issue in the presentation. Looking for your questions.
Thank you, Ying. A really excellent talk summarizing some really exciting advancements, I think, in proteomics. If there are any questions, please post them in the chat. We don't have a lot of time, but we have time for a couple of questions, I think. So I have a question to start with.
Yeah.
Ying, I was just wondering, you talked a lot about the sample preparation and the nanopods approaches, which really helps isolate those single cells and then prepare them for proteomics. But
Yeah.
do you think there are other advances that need to be implemented downstream of that? So in the chromatography and the mass spec itself, that would
Yeah,
help improve
that's a
those
great
numbers?
question. So as we know, the proteomics, single cell proteomics or spatial proteomics is still pretty early. So we see, not every step needs to be optimized. What we do in EMSO is try to improve the sample prep, including
Mm-hmm.
cell isolation, how we isolate the cell from the plant, and but really, if we want to get a high throughput. Now we are heavily limited by the throughput, how many cell, how many plant we can analyze. So usually we are at the 100 cell to 1,000 cell stage now, but still take a lot of effort to run all the cells because we do not have a good barcoding approach,
Right.
and also, the sensitivity of mass spectrometry still need to improve. So we can measure like not only 1,000 protein, we hope we can measure 5,000 protein to get a very deep coverage, similar to RNA sequencing, because we cannot unify the protein, right?
Okay, so we have a lot of questions now coming up in the Q&A. So the first question from Ken Birnbaum. He asks, for the plant cells, what is the complexity of the output data on mass spectrometry? For example, how much of the quantitative output is taken up by the top 5 or so abundant proteins? So
Yeah, that's a great question. So that's a way we also see the dynamic range is also one challenge of the plant, because plant is dominated by the several high abundant proteins, and so we address this one. Actually, this is already addressed in the mammalian research, because we do RC separation. So we separate all the high abundant species with the low abundant one, and all the mass spec also have improved the approach to measure high abundant and low abundant together. So still, we are not limited by the dimension right now. We are still limited by sensitivity. We still want to get high sensitivity to measure the low abundant one. So in terms of the output data, so we don't see the major difference between RNA sequencing and protein. We still get the abundance metrics. So but we see some difference in the RNA sequencing data, kind of the CV is high. Well, I don't show the data, but we see the protein is quite stable, because in theory most of protein should present in the cell. So protein is quite stable. You don't have the missing value. We do have missing, but the missing value is not a big problem for the protein. I mean, still the sensitivity and throughput is the major challenge for proteomics.
okay, and then Glenn Ur asks, what about making use of imaging mass specs to avoid the whole protoplasting?
So the imaging, if I understand the question correctly, is you mean the use the other technology, mass spec technology, like
MALDI.
MALDI? Yeah. So MALDI is very limited to measure protein. Most of MALDI is measure metabolites, lipids. It's very sensitive, but for protein, it's very challenging. And the reason is just you have to do digestion to separate all the peptide to run the samples. So imaging mass spectrometry, you just measure everything together. So you do have a problem with dynamic range, because you only see a high abundant one, because the ionization and suppression is the major limitation of imaging mass spectrometry for protein.
Okay, and then just very quickly, Peter Denoff asks, were you able to identify low expressed proteins like transcription factors in your data sets?
Yeah, that's the limitation. We only able to see the high abundant one, and but we do see transcription factor if they're high abundant. We only look at the high abundance of species, no matter what they are. So, but a lot of I do agree we need to increase more, make the mass spectrometry more sensitive to look at all these transcription factor, all the signaling pathways.
Okay, great. Thank you very much, Ying.